This is part 2 of the Chapter 4 Fungi lecture. All right, here we go. So we're going to start with habitat and metabolism for the fungi. Metabolism actually refers to all the chemical reactions that occur inside of the cells of an organism, but here it's mostly used to describe the nutrition. So fungi can live in habitats that are moist, slightly acidic. They can grow with or without light. Um, Most are obligate aerobes. That means they require oxygen to survive. There are species such as the chytrids or the Chytridiomycota. That's the phylum of one of the phyla of fungi that we're going to learn, that reside in the rumen of cattle. That means they live inside the intestines, the stomach. So the GI tract, we'll say it like that. And they're obligate anaerobes because that means that oxygen would actually kill them. Okay, so they carry out their respiration in the absence of oxygen. And yeast that's used to make bread and beer and alcoholic beverages are facultative anaerobes. That means that we do in Bio 111, we do an experiment. You may have done this, where we take yeast and we grow the yeast in the presence of oxygen, and then we take yeast and grow with in a container that is sealed. So whenever you do that, you get fermentation, which is anaerobic, and you get alcohol that's produced as a byproduct. All right, the reproduction of fungi is sexual and asexual. If they're perfect fungi, then they reproduce both sexually and asexually. If they're imperfect, they only reproduce asexually. In both sexual and asexual reproduction, fungi produce spores. So the reproduction is going to involve spores either way. And you can see here a puffball. You may have stepped on one of these. This one is a giant puffball. You may have stepped on one of these growing in your yard or in the woods, walking in the woods, and seeing the puff of, it looks almost like a puff of powder or smoke, but that's the spores that are being spread. Okay, so here we see a microscopic image photograph of yeast that are reproducing asexually by a process called budding. And this is one that you can see doing that. This little part is the offspring of that yeast cell is growing, like it's almost like a growth that grows on the side of, on one part of the yeast cell, and it'll get bigger and bigger, and then it will pinch off. So fungi can reproduce asexually by fragmentation, by budding like the yeast, or by producing spores. Fragments of hyphae can also grow new colonies. All right, this is the life cycle of a typical fungus. The asexual life cycle, we'll look at that first. We will start with the mycelium, which is haploid. The mycelium is just a collection of hyphae, remember. So this is one that has septa. It's a septated hyphae, where the nuclei are divided, by septa or walls. This mycelium, the nuclei of all of these cells are haploid, containing half the chromosomes. So what happens is they undergo mitosis and produce spores, and these spores would also be haploid. So in Bio 111, when we learned mitosis, we learned that a diploid parent divides and produces two identical diploid offspring, diploid daughter cells. But mitosis can also be haploid to haploid. So the haploid cells in the mycelium are dividing by mitosis. That means they're cloning themselves to produce haploid spores. And then the spores germinate, germinate means grow. They land on a moist surface, maybe on the forest floor, and then they start growing into hyphae, and eventually the collection of hyphae is called the mycelium. Now, for the sexual reproduction, you still starting with the mycelium that are haploid. They undergo something called plasmogamy. Plasmogamy is when haploid cells from two different mycelia fuse to form a heterokaryotic cell that has two or more nuclei. So this is the heterokaryotic stage, and it's showing one nucleus that's black and one that's red. So you can see that they're coming from two different mycelia. After the heterokaryotic stage, the nuclei undergo karyogamy. And karyogamy is when the nuclei fuse to form a diploid zygote. And that's what you have here. And this zygote is 2N. It's diploid. So it's just the fusion of the two nuclei from the heterokaryotic stage. The zygote undergoes meiosis to produce haploid spores. And then those haploid spores germinate into mycelium like they did in the asexual part of the life cycle. This is showing a fungus, is showing the hyphae, and then it's showing the sporangium. The sporangium at the end of a hyphae or a hypha is called a sporangiophore. So it says, this bright field light micrograph shows the release of spores from a sporangium at the end of a hypha, called a sporangiophore. This organism is a mucor fungus, which is a mold that's often found indoors. So this is the sporangium. And the hyphae that contains the sporangium is called the sporangiophore. And then the spores are released from the sporangium. Now, our first phylum of fungi. Fungi is the kingdom. So the different groups that we're going to learn are called phyla. So in the Protista, we learned the different groups were supergroups, but we're going to learn different phyla in the fungi. So the first phylum is the most primitive. That means it's the oldest, according to evolution. It's called the chytrids, or the Chytridia mycota. Most are unicellular. There are a few that are multicellular, but most are unicellular. Chytrids usually live in aquatic environments, although some live on land. Some are parasites, and others are saprobes, or saprophytes. All right, then we have the zygomycota, and the best example, or the most popular, I guess the most well-known example of zygomycota is the black bread mold, which can also form on fruit, like strawberries. But this is a picture of that black bread mold, and this is another picture of it. You can see the spores on the end. And they do normally reproduce asexually, so they're imperfect. This is the life cycle of a zygomycete, or an individual zygomycota. You can see that it can reproduce asexually, and most often it does, but it can also reproduce sexually. So we wouldn't call it imperfect because it can reproduce sexually. So if we look at the sexual reproduction, if we start with the spores, we know spores are haploid and they're released from the sporangium that are on the end of the hyphae. So in zygomycota, there are two different mating types. We don't have male and female, but we do have positive and negative mating types. And what happens is that plasmogamy occurs. That's the fusion between the positive and negative mating types. You can see here the positive mating types have red nuclei, and the negative mating types have blue nuclei. And the hyphae are aseptate. They don't have septa. So when the nuclei fuse between the two mating types, this produces a zygosporangium, which has many haploid nuclei. Unlike the life cycle that we looked at, just the generic fungi life cycle, there were just two nuclei. But here, there are many nuclei, so we would say the zygosporangium contains multiple haploid nuclei. This is the plasmogamy stage when the zygosporangium is fused. And then karyogamy occurs, and that's when the nuclei fuse. And that produces a zygote with multiple diploid nuclei. And the nuclei here are purple in the zygote. This is the zygote in the center. They're purple because that's what happens when you get red and blue. Red and blue make purple. So there are two nuclei, and both nuclei are diploid, not haploid. And then a sporangium grows on a short stalk, and haploid spores are formed inside. So the sporangium germinates or grows from the zygote. And the sporangium inside, or the sporangium that forms on the end, is haploid, and this occurs by meiosis. So that's the reason it's haploid. And the sporangium releases haploid spores. The Ascomycota are the next phylum. This is a phylum of fungi. So they're characterized because they form an ascus, which is a sac-like structure that contains haploid ascospores. They're just spores. They're called ascospores because they're found in the phylum Ascomycota. The life cycle of the Ascomycota can be asexual or sexual. The asexual is similar to all of the fungal life cycles that we've seen. The plasmogamy for the sexual reproduction, the plasmogamy that occurs. Okay, so sorry, there's still the positive and negative mating types. Now in the Ascomycota, they are the mycelia or the hyphae are septate. Trying to find here we go, my pointer. Okay, so we have the Ascogonium, which is from the positive mating type, or the ones with the red nuclei, and the Antheridium is from the negative mating type. Antheridium is a word that typically refers to the male part, and Ascogonium is the female part. So instead of positive and negative, we're having, more of a male and female parent. So the Ascogonium and the Antheridium fuse, and they contain multiple haploid nuclei. And when they fuse, that's called plasmogamy. And when they fuse, they undergo mitosis. And eventually, they form a structure called the ascocarp. Mitosis occurs in all of these cells here. And at the end of the ascocarp is an ascus. And the ascus contains one haploid nucleus from the Ascogonium and Antheridium. It says the nuclei in the asci, each one of these ascuses or asci, the nuclei fuse, and that produces a zygote. And the zygote is diploid. You can see the nucleus is purple because red and blue make purple. And the zygote undergoes meiosis, and the ascus now has 4 haploid nuclei. So those nuclei are haploid. Those haploid nuclei undergo mitosis. They double and form 8 ascospores, which are haploid again. And those ascospores will germinate into mycelia. The Basidiomycota is probably the most well-known of the fungi. The phylum Basidiomycota are the mushrooms. They have Basidio means they have a club-shaped fruiting body called a basidia. So this is the mushrooms and the bracket fungi, and the shelf fungi, the ones that grow like on the edge of the trunk of a tree. They look like little shelves. But these are mushrooms that you see growing in a circle. All right, they are only undergo sexual reproduction. And basically, you have 2 mating types, positive and negative. Plasmogamy is when the nuclei fuse between the 2 mating types, and that results in a dikaryotic mycelium. And dikaryotic simply means that each of the individual cells of the mycelium have 2 haploid nuclei, one from each of the mating types. That dikaryotic mycelium undergoes mitosis.